Good afternoon once again. My name is Jiří Urban, and I would like to really thank you, all of you, for coming for today's lecture. And before we start, I would like to introduce our today's speaker. It's Professor Albert Hek from Utrecht University in Netherlands, and he is expert in protein mass spectrometry, which is technology he applies to problems in proteomic and structural biology. He also established track record in quantitative proteomics and also protein post-translation modification studies, such as phosphorylation and glycosylation. And he also applies this technique to study, for example, host-pathogen interaction and stem cells differentiation. Professor Hek is also a leading scientist in native and cross-linking mass spectrometry fields, and he empowered the structure analysis of protein assemblies through these techniques. The group of Professor Hek also developed mass analyzer dedicated for this work and used them to study the structures and dynamics of these immune complex and viruses systems. And if you prefer numbers than letters, I would like to say that Professor Hek published almost 900 publications with 52,000 citations, and his H index is impressive number of 109. Professor Hek received several numbers of awards for his nice work, such as, for example, Gold Medal of the Dutch Royal Chemical Society, HUPO Discovery Award in Proteomic Sciences, the Spinoza Award, and the Krebs Medal. And he's also a member of Dutch Royal Academy of Science and Arts and the European Molecular Biology Organization. But there is still one medal missing in Professor Hek's portfolio. So I would like to invite Professor Hek to the stage, and I would like to present him MENDEL medal, which is play word, really. And so So with that, I would like to thank you again for coming, and I will give the word to the Professor Hek. Thank you very much.
Thank you very much for this introduction, and thank you all for coming here. I'm very honored and delighted to be here. This is such a historic place. My work is really focused on protein, so I actually sometimes make jokes about genetics, but I will not do today because this is holy ground. You know, we all have these expressions. I think general society, they all know what you mean if you say, oh, it's in your DNA or it's in your genes. I like to say today a little bit, it's in your blood, which in Dutch is also an expression. Is that the same? No, it's quite different. But I will come back to that in my lecture to show you that all of us have different blood, luckily. But also especially when I talk about these antibodies, they are really different in each of us. And I think that's very intriguing. And I hope to cross that message to you. Now, sometimes people say, what are you? Are you a biochemist? Are you an analytical chemist? Yeah, my shortest answer is I'm a mass spectrometrist. I was appointed years ago in Utrecht. The chair that I have is called Biomolecular Mass Spectrometry and Proteomics. Only people who do it can pronounce that. But it really covers what I do. And it's, for me, has been a great time to be in that field because mass spectrometry over the years has maybe become more and more important in the life sciences. And by being maybe a nerd in this technology, you get associated and you get to work with many people in many different fields, as Jiří said, in stem cell biology, in virology, in many different fields. And I feel very privileged that I, through that collaborations and through that work, know a lot about a lot of things, but actually I mostly say I know very little about a lot of things. And so also about antibodies, I'm not an expert. I'm just getting into this from mass spectrometry. But I hope to show you that we discover things that were not discovered before because you use a new technology that was not used for that method yet. And I think this is where science is always going. If you try something new, you will discover unexplored arenas, and that's what I hope I can show you today. So, yeah, a little bit about genetics. That's sort of what I said. Of course, you have to show this slide here, maybe. I think I don't have to explain this, maybe to myself, but I think you all know it. But, of course, we all know that it is in the genes, and we know about how they divide and how they can be dominant or not dominant. But what about the genes that make our antibodies? I will come back to that in a second as well. Now, first, I also want to show something as an acknowledgement, and here he said about how many papers I published. Now, that's not true. I was at Utrecht University this year for 25 years, and we had this great occasion that all the alumni from the group came back to Utrecht. So I have now this beautiful picture. This is not my group, but it has been my group. So these are all the people that worked in the group over the last 25 years, maybe not all. And there are also many friends of the group that are also on this picture, like people like Matthias Mann and Rudy Ebersold and Alexander Makarov. So we had a big party in April this year. But I show this slide also because whatever I show and whatever medals I get, it's really due to the people behind the science. And it was great to give them something back because we had a symposium, but we also had a great party. Now, as a mass spectrometrist, and also just a little bit as an introduction to me and our group, is a timeline that people made when we had this party. And it shows that I arrived in Utrecht in 1998. The word proteomics was just barely surfacing. And yeah, in my job as a professor, I really started to work on what we call structural mass spectrometry and proteomics. And this was a sort of parallel development of things we did. I think we were maybe the first, or at least one of the first, to do phospho-enrichment with titanium dioxide. But we also came with new isotope labeling methods. But at the same time, we got a niche in the field of mass spectrometry, analyzing not small molecules, not peptides, but analyzing whole proteins. And we took that a step further, and we analyzed whole viruses. And so we can now mass analyze whatever we want to mass analyze, if they are not glycoconjugates, I have to say to someone in the room here. But you know, this development of technology brought us further into our applications. Now, I will not talk about all this. I just want to focus on this last part where we are now, where we got really into this research on antibodies. But what I still want to say, we only got there because we developed new methods to analyze antibodies, also to do mass spectrometry on single molecules. So this is about 1 million times more sensitive than we could do before. Also on getting rid of DNA. Can we do de novo sequencing? Can we read off sequences of proteins without a DNA template? And also some other techniques that I mentioned over here. But that accumulated in what I'm going to talk about now. What is your antibody repertoire? Can we find antibodies of interest? Which antibodies are you making at the moment? If you had corona again a few weeks ago or so, you're probably making antibodies against corona at the moment. If you had a flu or something else, you're making antibodies against that. So your body is constantly making antibodies. Many of them end up in your blood. Can we study those, and can we find maybe antibodies of interest? And then when we do this, yeah, we sometimes stumble upon something that is so new and so against all the textbooks that you think, how can this be possible? And in the end, if we have time, I also want to show an example of that, where we sort of redefine the structure of a molecule that is in all the immunology textbooks, IgM, but it turns out to be wrong in all the IgM textbooks. So that's something that I hope I can come to as well. Now, what I'm not going to talk about, we also do a bit. There's a big movement now in mass spectrometry and mass spectrometry-based proteomics to measure as many proteins as possible, for instance, in someone's serum. And this can be done now relatively cost-effective. So you get a few drops of blood, And within an hour or so, you can measure the level of all the proteins in the blood. Now, you can say, why do you want to do this? Now, yeah, that means that you can really do multi-assays on any biomarker in your blood and directly monitor if something goes up or down. And in this way, you can maybe see how we respond to drugs or how we maybe have a time point of inflammation. And this is really now booming, and a lot of research groups are doing this sort of efforts, this blood-based plasma proteomics. So this is a big thing, but you always have to be careful to be too enthusiastic, because if you are like that, you start a company, you get billions, and then you get into jail. So what I just said, take it with a little pinch of salt, but I still think this is an interesting way forward, doing serum or plasma proteomics. But if you look at serum or your blood, there's a lot of proteins in there. On the other hand, it's a very strange composition of proteins, and I show that a little bit on this pie chart. So this pie chart shows all the proteins in your blood, but based on their relative ratio in protein concentration in your blood. So you directly see that about, 1/3 of all the proteins in your blood is albumin. But you also see, and that's where I want to focus on, you see that this part, IgG1, but there's also here IgA1 and IgM1, these immunoglobulins or antibodies, yeah, they are also about 1/3 or maybe even more of all the proteins in your blood. Now, in most of plasma proteomics, people say, oh, albumin is not interesting, I'm not looking at it, I want to go to the low abundant biomarkers. Also, most people say, let's not look at the immunoglobulins, but we don't know what to make of them. So they are typically disregarded in proteomic studies. Now, if you do that, you disregard, 70% of the proteins in your blood. So what we do is we said, can we really look at the antibodies in our blood, and can we learn about them, and what are they? Now, just a little bit of numbers. You and I have about 4 to 6 liters of blood in our body. And in this blood, there's about 30 grams of immunoglobulin 1. So this is quite a huge amount if you calculate in molecules. And what we do know is that almost every 3 weeks, every molecule of antibody is degraded and is built again. So you might say in 3 weeks you have a total new set of antibodies, because everything has been degraded, everything has been built again. Are these the same antibodies? Are these different antibodies? That's a question that we want to have a look at. Now, this is my textbook about antibodies, and I hope you all are still with me here. We have all kind of different antibodies. So they are subclasses, IgG, IgA, IgM. And you see cartoons at the top. We all think that an antibody is a Y-shaped molecule. I don't know why, but the textbook says so. And we know that in IgG we also have these subclasses, IgG1, IgG2, IgG3, IgG4. IgA is different. It can be monomeric, it can also be dimeric. And IgM is also very special. It can be pentameric or hexameric. Now, these are just cartoons. If you go a little bit more in detail, which is at the bottom here, then we know that an IgG is built up of 2 heavy chains and 2 light chains. And the way I draw these cartoons here is that in these heavy chains you have this black part, which I now call the boring part. It's called the constant region. Then at the top part, both in the light chain and the heavy chain, you have this orange region, which is not orange because it's Dutch, but it's orange because it contains these hypervariable regions. So there are 3 hypervariable regions in the light chain and 3 hypervariable regions in the heavy chain. Then the subclasses are slightly different from each other by the blue things, which are the disulfide bridges, the amount of disulfide bridges or the length of the hinge. And this is all contained in this IgG. Now, IgA is slightly different. It also has O-glycosylations here. There's also something else on this cartoon that I have to explain. Bacteria don't like our antibodies. The first thing they want to do is kill our antibodies, because then they can survive better. So they make proteases, like this IDS, and here this OGPA, and what they do is they specifically attack antibodies, and they cleave these antibodies by cleaving them here in the hinge. And by cleaving in the hinge, we can use this in our method, because by cleaving in the hinge, we can actually take this part away from that part. In this way, we lose the glycosylated part, and we are only stuck with the variable part. And we are really interested in this variable part. I will come back to that later. So there are all these subclasses with their small differences. Also biologically, they have different roles, the different antibodies. Even the subclasses have different roles. But I argue we don't fully understand yet what the differences all are. Now comes the thing. How many antibodies can we make? And here the genetics is still true. Mendel is still right. But it's quite complicated. So I think you probably all know that in these variable regions of the antibodies, they are actually made of segments of genes, V, D, and J genes. And they are sort of redundant. 3 of here, there are 6, 7 of here, 5 of there. And when these genes are made, they can shuffle and take once this V gene, and then this J gene, and then this D gene. And they can make all these combinatorial possibilities. They can do this in the heavy chain, but they can also do this in the light chain. And through this, you don't get one gene, but you get combinatorial combinations. If there were 2 segments, you could have, like Mendel, an AA, a large A, and 2 large A's. But if you have so many as here, you can actually calculate that theoretically, you can make 10 to the 18th different antibody gene sequences. Now, I don't know what this number means, 10 to the 18th. Now, I can tell you you can't, because the antibodies need to be made by cells. And they are exclusively made by B cells. Now, in our body, and you know, I like numbers, in our body, we are quite limited. We only have 10 to the 13 cells. So that means that we cannot make 10 to the 18 different antibodies. We only have 10 to the 13 cells, about 1%, this is an estimate, of those are B cells. And only 1% of those, again an estimate, are B cells that are really secreting antibody. So luckily this 10 to the 18th is completely wrong. It's a much smaller number, it's only 10 to the 9th. So still, you can make 10 to the 9th different antibodies. That's a number I want you to remember. Now I come in as a mass spectrometrist. What does that mean? And it's a complicated slide, but what I want to show you here is this combinatorial recombination of all these different genes. And they all lead to an antibody sequence that you see here at the bottom, where certain parts are very constant. So every antibody in your body, in its kappa chain, has here a threonine, a serine, a proline, no variation. But in this part, which is one of these CDR parts, it's hypervariable. It's very small because it can be almost any amino acid. And then you have a hypervariable part here, which is very small here, another hypervariable part here. So this is sort of taking the consensus sequence of about 1 million sequenced antibodies. And so these sequenced antibodies, they have positions that they have all in common. I would say boring. And they have positions that are hypervariable. And these hypervariable regions are really essential because they form the recognizing part that can act against the pathogen. So the specificity of the antibody comes completely from having different sequences in these variable parts. So it's very important to know these variable parts, while it's not so important to know the rest, because we know the rest already. Yeah, so how do we know these sequences of antibodies? Now, what you can do is use genetics, and this is how it's mostly done. So you can try to get all the B cells from your body, and then you can take the B cell receptor, you can use sequencing methods, RNA-seq or whole genome sequencing. There's all kind of methods now possible. And then you can get the sequences of all the B cells. Now, there's a big caveat there. Which B cells can you get? Now, you can easily get the B cells that are circulating in your blood. But it's well known that the antibodies that are produced and come in your blood are not made in your blood. No, they come from B cells that are in your bone marrow or in your lymph node. And although many of you would volunteer to give a bit of blood, not many of you would volunteer to give B cells from a bone marrow or from a lymph node. So it's actually quite hard to get the B cell sequences. The other problem is, even if you have 1 million sequences, or even more, it doesn't tell you yet which of these B cells is really on and producing and secreting antibodies that end up in circulation. Yeah, so this is the method of choice so far. It works, but it doesn't tell you which antibodies are really made and end up in circulation. Now, then comes our big breakthrough. Oh, but it looks, with hindsight, extremely simple. So what do? We just take a few drops of blood. I think 10 microliters is sufficient. And we purify with it's a nanobody. We purify all the IgG from the blood. So then we have all the IgG bound by its Fc tail, so at the bottom. And then we come in with one of these bacterial proteases, and that bacterial protease cleaves here just above the hinge. So here's the affinity matrix. We capture all IgGs, independent of their sequence. But then we cleave it off here, and then we isolate what we call the FAB arms. This is the FAB fragment of the antibody. It contains all these variable regions. Now, the first person who I asked this to do was actually Czech. His name was Vojtěch Franč. And he said to me, no way. If I do this, I get 10 to the 9th different sequences. I forgot to tell, and I'm a real mass spectrometrist, so I need to go back. All these sequences have a slightly different mass. They almost have all the same mass, of about 47,000. But depending if you have a valine or an alanine or a glycine or a tryptophan at a certain position, it changes your mass. So all FAB arms will have a slightly different mass. That's what I forgot to tell, and I need to tell you. Okay, what do? We capture all the IgG. We shave off the FABs. These are these 45 to 50 kilodalton fragments. And now we do simple mass spectrometry, LC-MS. We bring all these FAB fragments to the mass spectrometer and we measure their mass. Now, the hypothesis was that we would get a big blob with a lot of masses that we could not interpret. Now, the strange thing came when we did this the first time, and this was the LC-MS chromatogram of the data. And I like to show this. It's not a beautiful figure, but it's the original one. And today I enjoyed how nice it is to see original ones. But this is our original LC-MS. It looks, for people who are in proteomics, almost like a triptych digest of BSA. It looks very simple. There are a few dominating peaks, maybe 100 or 200, but it doesn't look like there are 10 to the 9th or 10 to the 7th or 10 to the whatever peaks there. Now what we can do now, and this is my, no, I want to get the animation going, but let me see if I can do it like this. So what we do now is we scale over this LC-MS chromatogram, and then on the right we measure the masses. And they are always 48, 47,000, but they are always slightly different. So we measure here the mass of every FAB molecule, and we measure the retention time of every FAB molecule. Now, first of all, and I discussed this with Jiri a bit, I'm surprised that we can separate all these FAB molecules by LC-MS, but through mass spectrometry, we can now measure all these masses as well. Now, what do I know now about a specific FAB clone, as we call it? I know its mass and I know its retention time. Now, what I can do now, I can convert these LC-MS data into a plot like this, which we now call an antibody repertoire. Now, what do you see here? You see some peaks. They all have a mass between 46,000 and 50,000. And you also see an intensity. And that intensity is scaled because we also put in a few synthetic recombinant antibodies. So you directly see here the concentration of each of these clones that we detect. And nothing of this is noise. They are all clones. But some of them are way more abundant than some others. So this is what we call the blood antibody repertoire of a given person. The antibodies that are present in the blood. Now then we started to do follow-up experiments and we said, okay, let's take the blood of a person, but let's also take it a month later and maybe a month later again. And what I told you, in 2 or 3 weeks, every antibody is broken down and maybe replenished. Now what you see in this data is that the antibody profile in red doesn't change over a period of 2 months. So that means that in your blood, your antibodies are identical now and in 2 months, although they are all new. And they are all newly synthesized. So you sort of make the same antibodies all the time. Now we detect, and that's the number in the middle, about 500 clones. There are many more, but this is sort of our detection limit. But we see that, and that's in red, about the top 30 covers already 50% of the concentration. That's how we make these plots now. Now at the bottom, you see another person, another antibody profile. And again, it's the same. It doesn't change over time. Also here, a couple of antibodies are very dominant. They take up 80-90% of all the antibodies in our blood. And now the most remarkable, if we compare these two plots, the overlap is 0. So you have a constant antibody profile, but it's totally different from your neighbor. Yeah, so everyone has a unique profile. Now, of course, this was our first experiment. We said, okay, this is very exciting. No one has really noticed this before. But what does it now really mean? And it's only 2 people. So we were sort of lucky and unlucky. This is when COVID started. But we got in contact with people in Greifswald from Barbara Breuker. And she had a cohort that I really liked. It's also only 10 people. But blood was taken at, what you see here, 9 time points. And also these people got a vaccination, a COVID vaccination, 3 times. And so each time we got blood just prior to the next vaccination. Now that the vaccination worked, we could measure by measuring the titer against SARS-CoV-2 spike protein. And indeed you see that after vaccination the titers go up. Then there's a long time that we didn't do anything, and here was then the booster vaccination, and then you see the titers going up again. So yes, people are making new antibodies induced by the vaccine, so that's good. Now for us this was a nice cohort because it extended from 2 months to a year, and we have 9 time points for every person. So I'm going to show a few examples of our data now, just to start with one. So this is one donor, and maybe you don't see the colors, and maybe you don't even see the figure, but you just have to believe me, they look 9 times identical. So everything that's in this yellow ochreish color is identical. Everything that's black is new, that we don't see at time point 0. So basically this confirms our earlier data, that even in a year time, your antibody repertoire doesn't change that much. And even if you get vaccinated 3 times, your antibody repertoire of dominant antibodies in your blood doesn't change that much. Now again, this is one donor. We have this now for 11 donors. Some show a little bit more striking, like this donor, I think it's donor 13, shows a very abundant antibody that we didn't detect in the first months, but is sort of dominating at the end. This repertoire, you also see, indeed, there are some new antibodies coming up, but it's also really remarkable that the most abundant antibodies in your blood, they remain very stable for a very long time. What does that mean? We also say antibodies are very good, and B cells are very good in making mutations to mature the antibody, to make it better. This means, because of we having exactly the same mass, that these are antibody sequences that are not mutated at all over a year time. So it's really B cells that don't further mature, but keep making the same antibody, in this case for nearly a year, even when they are challenged with a vaccination. Now, to show this data differently, we can really look at all the data. We can make a hierarchical clustering. And I almost don't dare to say it, but it's almost like a DNA vignette of every person. So you see that every person has an antibody repertoire that really matches very well with their own antibody repertoire over a year time, while some, while the differences between people are wild. We see no overlap at all. Now what is nice here, we see one person that was a mismatch. It should have been donor 19, but I don't know, a nurse or a PhD student screw up. But we didn't know this, but we directly see this cannot be true. This is an antibody repertoire that doesn't make any match with any of the other antibody repertoires. A sample got mixed up. Some, you see no difference at all, while some you see a bigger difference. The bigger this line, the bigger the differences is. And if we see a difference, it's often between the last time points, 7, 8, and 9, and here also 7, 8, and 9, and the other time points. So that's with 6 months in between. So, but out of all these donors, there are maybe 2, donor 5 and donor 13, that show the biggest variation, but all the others don't show that much variation at all. Now, to look a little bit further into that, here we sort of compared in these pie charts, does your antibody repertoire still look the same after a year? If it's totally green, it's identical. And the more red it is, the more it's different after a year. So we see some differences, but mostly they are green, but there are a few donors that are very red. If we then go to these red donors, then it turns out that there are specific clones that we identify by their mass and unique mass and retention time that come up or go down at these late time points. So that gives us an idea that you make your own set of antibodies. They are unique, but of course we have many more antibodies in our blood than these couple of hundreds that we detect. So the ones that we detect, and this is a general problem in proteomics, are the most abundant ones. Yeah, so are there more antibodies there? Why don't you see a difference induced by the vaccination? Maybe because the antibodies made upon vaccination are lower abundant than the ones that we detect. Can we work on that? Now what we can also do is what we call now, we can measure the full antibody repertoire in blood, but we can also detect what we call the antigen-specific repertoire. So the experiment we do is we couple the whole spike protein of corona to a bead. We fish first for the antibodies that bind against the spike. Then we do the same trick and we analyze the profile of these antibodies. Now the nice thing here is that we have this spike protein, but we can also make the spike proteins of the different variants. So what was called the Wuhan variant or the South African variant. We can all couple them separately to the beads, and that's an experiment we did. But first, this experiment, we see that this is the normal plasma repertoire of this donor. If we then go to the repertoire against the spike protein, we see much less peaks, still about 20, 30. So against the spike, this person makes 30 abundant IgG antibodies. They are way less abundant than those. It's also multiplied by 10 compared to here. But there is a pattern of about 10, 20 antibodies that this person makes against this, let's call it Wuhan spike protein. Now then you can continue, and that's what we also did. I have to show there, and again it's a full slide. But what we did is we used different variants of the spike protein representing the different viruses. And you see that all these spike proteins, they pull down sort of a similar subset of clones, antibody clones, that react against the spike protein. But you also see some differences, like this brown one over here. It's totally absent if we use these two, while this one seems to be present all the time. Now, what does that mean? It means that this antibody is cross-reactive. And no matter which virus variant you got, this antibody could do the job. While this antibody here, that you also made, or this donor made, is only working against these two variants, but is not working against these two variants. So you see that you make this mixture of antibodies against the virus, and they all have different epitopes on how they bind. They all recognize the virus particle differently, and that's why some of us are maybe more protected against new variants than others of us. And so with this work, we can show that we can go deeper into the antibody repertoire, more to the antigen-directed repertoire, but we can also learn something about how we respond. You know, immunology-wise, you would say, it's stupid if I would only make one antibody against the spike. Make as many as you can, because if one fails, the others can take over. Now, that's a bit what you see here. We really see that with clonal resolution, and if you blow this up, you actually see maybe 50 different peaks. You make a whole array of different sequences. They are not similar, they are very different, but they all have the ability to bind to the spike protein. And then, if we do this with, again, I think it was 8 donors, we find for each donor about 50 clones that bind against spike. If we compare these clones with each other, each of us makes another repertoire of anti-spike antibodies. So who of you made the best sequence to be developed in a therapeutic? I have no idea, but I'm pretty sure that some of you did. And maybe it would have been the one that is really cross-reactive against any variants, and that maybe also is very good and that we don't study here in neutralizing the virus. But the message I want to get across, all of us react differently. We all make antibodies. They all bind against the pathogen, neutralize the virus, hopefully, but we all do this in a slightly different way. Now, we can go wild now, because this was antibodies against spike, but we can now fish for any antibody response. So this is just one other example where we look at autoantibodies involved in rheumatoid arthritis, where proteins get citrinylated, and we can actually fish for the antibodies that specifically bind against the antigen of concern, which is here citrinylated peptide. Now, if we do that in RA patients, again, we find that every RA patient has a repertoire, and here our methods got slightly better, so we get really up to 1,000 different clones. But again, we see that every rheumatoid arthritis person is unique in the autoantibody profile that they make against these autoantigens. And yeah, that I don't know what that means on how we should treat all these people, but it clearly shows that this is a level of antibody knowledge that I think we didn't know so far. So that brings me to the end of the first part. What did I show and what I didn't show? And I was lucky to get funded by the ERC for this work. And what did I promise? So I tell you now, so I have to deliver. Most of the work I showed is really about IgG1, because we have a specific bait for IgG1. We have a specific protease that cleaves off the FABs of IgG1. We are not exploring yet all the other ones, IgG2, IgG3, IgG4, IgA, IgM. We can, because they are also antibodies in your blood, but we need special methods for that as well, and we will develop that. I showed you, you know, antibodies in your blood, but there's also antibodies in your saliva, in breast milk that you give to your babies, very important. It's full of antibodies to protect the baby, and we can also use those fluids, and we have shown that we can use these fluids as well. We can also work on antibody-specific repertoires, as I told you, and there we can use, you know, any virus, but also any bacteria, or maybe even cancer-related mutations, if there are autoantibodies or antibodies made against that. Now, the big bottleneck, and for me as a mass spectrometrist, is, okay, you tell this antibody has this mass, but what is its sequence? And there we are developing methods, and I'm not going to tell about this today, can we really sequence every one of these clones that I mentioned and get the CDR regions? If we have that, then we can make the protein recombinantly, and we can really test that functionally. Can we do this? The simple answer is yes, we can do it for some of the really abundant antibodies that I showed you. Can we do it for all of them? Someone at the ERC thought maybe he can, because they gave me the money. But this is still going to be a lot of work. It's very difficult because they all look so much alike. And having false identifications, false positives, is something that is very difficult. Yeah, I should disclose that we also started a company for that as well, to see if we could bring that further. So I want to shift gear now a little bit and talk in the second part more from the structural part. So, and it's also about antibodies, and it's about IgM. Now, this is also a model of IgM, as you see over here. It's this pentamer, there's this pink thingy over here, and some people say there might also be IgM hexamers that doesn't have this pink thing, and they are way more symmetric than these pentamers. So I think that's a bit the textbook knowledge. What does it look like, these pentamers and hexamers? They contain again this IgG-like protomer, as you called it, again with a heavy chain and a light chain. IgM is more complicated, it has many more glycosylation sites. And then 5 or 6 of these form this huge structure, and all these black lines are then disulfide bridges in these structures. Now, this is an important molecule. It's the first antibody that you make in response to a pathogen. So it's also been studied quite a bit. And here you see, for instance, some older, not even that old, EM pictures of this IgM. And it really shows that this pentamer is almost like the hexamer, but forgotten to make the last one. So it really has a hole, but still it has a sort of 6-fold symmetry. So that was the textbook knowledge. Also more recently, very high resolution structures came out for IgM. This was also always very difficult to do. But now they came out, and yeah, it gives way more information about IgM, and you know, they were all published in Nature, so they must be true, some people say, and some other people say not. But they all said that they were measuring IgM as present in blood, but they all missed something. And so I'm going to show what they missed. So how did this work? First, we just were doing the boring thing that I showed on slide 2, plasma proteomics. We just measured across hundreds of people. We measured all the levels of proteins in your blood. And we found in a very small cohort of about 50 people or so, we found that the concentration of IgM in your blood, when we compared it to this protein called CD5L, showed a nearly perfect correlation. So if your IgM is high, your CD5L is high. If your IgM is low, your CD5L is low. We thought, how can that be? And why, if you look up this protein, CD5L, not much is known about IgM and et cetera, et cetera. It has hundreds of papers about the role of CD5L, but nothing really saying that it does something with IgM, a few exceptions there. So this is only 40 people, so we also went to other data. This is 700 people. And again, you see overlaid in gray and red the concentration of IgM and the concentration of CD5L. And you really see, you know, when IgM goes up, CD5L goes up, etc. So this is a correlation, correlative evidence that they may have to do something with each other. So that was sort of the start. Yeah, what is this CD5L? Yeah, then you look up literature. It's known as a scavenger receptor expressed by macrophages, which is already weird, because IgM is expressed by B cells. It's remarkably understudied. There have been some papers where they suggest that CD5L can be carried by IgM, and there was even a little bit of an EM structure where they showed that the CD5L might bind in this pocket that is sort of empty in that hole of IgM. But what we now did is, again, a very simple experiment. We did size exclusion chromatography on whole blood, And then we looked where proteins co-elude in size exclusion chromatography. Now, for those that you don't know size exclusion chromatography, if there are very big protein complexes of 1 million Dalton, they elute here. If they are very small proteins, they elute here at the end. CD5L is a very small protein, so it should elute here. A J chain is even a smaller protein, it should also elute here. Now, if you look at the patterns of elution, then you see that here the elution of IgM, here the elution of the J chain, and here the elution of CD5L. And you see that these elution patterns are identical. So the whole of CD5L is not eluting here, but is really eluting together with IgM. So this really shows evidence that it is one complex. Now, for those of you that are very sharp, you say, hey, but the J chain has actually 2 maxima. Yeah, this is the J chain in IgM, and this is the J chain in IgA. So that makes sense that it actually has these 2 peaks. So this gives us evidence that it's really co-eluting, that it's maybe one immune complex, which was relatively new at that moment. And then we took us a year to make an antibody that recognizes CD5L. That was easy. But then we needed also an antibody that recognizes CD5L only when it's bound to IgM. And that was a bit more tricky. But finally we had one. And we could show that really every IgM molecule in your blood contains also CD5L. And then you ask, how can this have been overlooked? But this is sometimes how science works, I think. So, but we could really get evidence that every IgM molecule in your blood contains a CD5L. We also could show that they are really equimolar, CD5L and IgM, so there's really one CD5L molecule per IgM. Now a little bit of mass spectrometry. What we wanted to do next is measure the mass of this IgM CD5L J chain complex. And yeah, we had a unique tool which we developed a few years ago, and I'm really excited about, so I use a second to use this. We worked a long time just to measure whole viruses by mass spectrometry. And this is a virus called Flockhouse virus. It has a mass of about 10 million Dalton, but we still get an electrospace spectrum with a charge state distribution. But this is the virus with 165 charges, and you get a charge distribution, and maybe if you follow the course in mass spectrometry, you can take these peaks to calculate the mass, the charge. And if you do that, then you get to this mass of 9.3656 million Dalton plus or minus 1.5 Dalton. But what we did see in our Orbitrap, if we really brought the signal down of the molecules that we get into the Orbitrap, here we have a couple of million ions. But if we really bring it down to almost no ions, we see that the signal gets quantized. And that means that this is one ion or one particle. This is another ion. This is another ion. And here there are 2 ions with the same M over Z, or nearly the same M over Z. So we see twice the signal. Now, how big is that signal? Now, that signal is directly determined in the Orbitrap by the charge. So we do a charge measurement here, and this is just 4 ions, so maybe you don't believe me yet. But what we did now is we did this for a whole range of proteins that we could all do native mass spectrometry on, and we looked at the signal intensity of a single ion of all these particles. And the smaller the charge, the less the signal, but we really came to a calibration where we can independently, just from the intensity, measure the charge, but at the same time we also measure the M over Z. So that means that for every ion we have the M over Z and the Z, and that equation, you know, I can even solve, then we know directly what the mass is of the molecule. So we call this charge detection mass spectrometry. It has the advantage that we don't have to resolve these Gaussian charge distributions anymore. Now maybe this is too technical, but it really helped us in the IgM field. Just a little bit more, the way we do this now is that we detect a couple of hundred of these individual ions. We can filter out which ones are surviving in the Orbitrap, and then we can get really things like this where we see the charge going down, that's why it's going down a little bit, and we see and we can extract 2-dimensional mass spectra. This I wanted to show because we use it now to measure the mass of IgM, which normally is very heterogeneous molecule, but we were able to do this, and we were able to show that if you recombinantly express IgM without the J chain, the system goes berserk. And it forms pentamers, but it also forms hexamers, it also forms tetramers, and even some trimers. And so if you don't have the J chain, IgM oligomerizes, but very unspecifically. If you co-express it in the B cell with the J chain, all this disappears, and you get perfectly just one distribution, and this is IgM pentamer with a J chain with a mass of 942. So we were very happy, but now came the big question: what can we see if can we measure the difference between IgMJ and IgMJ CD5L? And maybe it's not that visible, but yes, we can. We really see a mass shift of about 40 kilodalton, and this CD5L is 38 kilodalton. So we really have direct evidence from this charge detection mass spectrometry that there's really CD5L in this complex. Now this was on recombinant antibody. Next, we also did it on purified IgM from serum of individual donors. And each time we only see one particle, and that is really the particle with IgM, J chain, and CD5L. So this is sort of the breakthrough. Every IgM molecule in your body is a pentamer, but it contains a J chain and it contains a CD5L. This was not known, and then of course the question is, why is it there? What does it do? Now I come back to something that I still don't understand at all. So IgM J chain is produced by B cells. The CD5L is not produced in the same B cell. So the CD5L is produced by other cells, and somewhere in our body they need to find each other, and the CD5L is incorporated in the IgM, and possibly only then it can really get into the bloodstream, is a hypothesis. So we showed that this is really not expressed in B cells. That was done over here. We also showed that we can express the IgM with recombinant CD5L. So we can do the experiment where we make recombinantly IgM with J chain. Then we can add CD5L, and then by using a bit tricks, a bit reducing conditions, we can get the incorporation of CD5L. So we show in vitro that CD5L can be later built into the IgM-J complex. But then the next question is, how is it bound? Now we showed by mutational analysis, where we could really say by mutations that this cysteine in the IgM and this cysteine in CD5L is really important for this. And this is not a structure, this is a structural model, thanks to AlphaFold, and we could sort of place this CD5L in the IgM. Then the next question is still, okay, it's there, does it matter? Now then you're going to look, what does IgM really do? Now it does a lot of things. It can bind to receptors. It can also activate complement. That's one of its main functions. And this was a bit of a disappointed experiment. We could produce IgM with CD5L or without CD5L, and it activated complement equally. That you could still call an experiment go wrong, but this is biology, so CD5L doesn't matter for complement activation. It is not needed to be present there. But also IgM needs to bind to its receptor. There are two receptors actually, FcU alpha R and FcUR. And again, with FcUR we see no difference, but with the top one receptor we really see a reduction of binding when CD5L is there. So there it may play a role. I don't have a full answer yet, but it seems that there are differences in the two molecules. Now another one that is really triggering something that we did next, we also looked at another molecule that secretory IgA, but also secretory IgM binds to, and that's a molecule called PIGR. And here we also showed that when the CD5L is present, the IgM cannot bind to PIGR anymore. Now for IgA, PIGR binding is extremely important because that's how the IgA can actually be transferred to the breast milk or to other saliva, to other secretory fluids, or even the mucosa. So binding to PIGR is extremely important for getting into that sort of fluids. So if CD5L is there, it seems that IgM cannot do that anymore. So the final experiments that we sort of did is we said, okay, let's now look at IgM in milk and in saliva. And to our surprise, and these are maybe complicated to read, but there is no CD5L in milk or saliva. So IgM in saliva or milk is totally devoid of CD5L. And instead, and I think that's the sort of final slide, instead it binds to PIGR. So when you talk, and that's I hope what stays here, if you talk now about IgM in our body, there are two unique IgM compartments. The B cell probably produces this, but then when it goes to secretory IgM, it binds to PIGR, and that's the complex that you see over here. And if it goes to circulatory IgM, like in your blood, then it doesn't bind to PIGR, it binds completely to CD5L, and all the blood molecules are looking like this. So again, it's a sort of new feature about antibodies that we didn't know yet. There's still a lot of questions that we need to address. How is it formed? Where is this CD5L produced? How does it get to the IgM? Why can it be so efficient? You know, is this one here first and can it exchange for CD5L or the other way around? These are things that we don't know yet. But with that, I hope I told you some interesting things about antibodies that you didn't know yet. I'm intrigued by this sort of work. I hope I could get you enthusiastic for it as well. I like to finish with acknowledging a lot of people through this work. Again, I'm a mass spectrometrist, but I work more and more with people in medical hospitals for the serum samples or other clinical samples. With that, I like to thank all the people that you see over here. This is the current group in Utrecht, and here's very prominently someone, so that must be a Czech person, which is true, and there are a few, Jan Fiala is on this picture somewhere as well. Now with that, I just want to say again, it's great honor to be here. It's great honor that you all showed up for my talk. And I hope I learned you something that you found intriguing. And with that, I'd like to thank you all for listening.
Thank you very much for a very nice and interesting talk. And the talk is now open for discussion. So if you have any question, let me know, and please wait for microphone.
Keep him running.
Yeah. Thank you very much for the talk. I have a question for the first part regarding the autoimmunity. So do you think it will be feasible, based on your mass spec data, maybe the sequencing and tools like AlphaFold, to design and produce maybe some neutralizing antibodies for patients with autoimmunity, like tailor-made for them?
This is difficult for me to answer, because I told you already we started a company, and there we say yes. And I also think the answer is yes, but I can also talk for an hour about what the problems still are. You know, if you want to sequence antibodies and you don't have genetic templates, and we don't, even if you have B-cell sequences of the same patients, it's not the right template for what we are seeing as antibodies. So you really have to do it with mass spec. Now, there are all kinds of problems. Leucine and an isoleucine by mass spec is very hard to discriminate. They have exactly the same mass. But there's also, you need complete sequence coverage because, and I forgot now, but it might also be that 2 amino acids have the same mass as 2 other amino acids. So you really need the sequence of every backbone. Now, I can tell you, we have a nice butterfly here, who maybe also has a question. So I can tell you, yes, we can, and we are sometimes successful already. We have our first successes, but I also should be realistic. No, it's still a long way, and my goal is to do this on all these couple of hundreds that come by, And we are also developing new mass spectrometers to do this. So yes, it will be possible, and especially also with AI and you know, with the recognition of features in our spectra. I think it will all be possible, but I cannot promise it will be possible tomorrow.
Thank you. Any other question?
So thank you for the nice presentation. I would ask about the first picture where you connected the CD5L and the IgM. In your big data, do you have a program that is looking for these correlations, or do you have to go one by one, you and your team, and just check what is interesting?
I like this question, because you should ask this to the students in the group, because this old guy does it one by one in an Excel file, and they write an R script, and they do it 100 times faster than I do. So yes, we have scripts that do this, and we see many more co-correlations in blood levels in donors. And I'm actually it's a completely different talk where I'm really interested in what's the normal level of a protein in your blood. And we see, for instance, that there we can really see that this is, for some proteins, all of us have the same concentration. For other proteins, you might have 5 times more than me. And what does that mean? So yes, we can do that sort of, and we are doing that sort of analysis. And then the CD5L IgM is just one of the correlations that we found in such data.
Next question.
So how much this uniqueness, like these patterns of each of us can be affected when you get like transfusion, for example?
Yeah, a perfect question. These experiments are underway. We first did another experiment, which I can tell because it sort of failed. So many people get rituximab, for instance, for treatment in autoimmune, like also arthritis and rheumatoid arthritis. And people and clinicians came to me, and I was very naive, and they said, oh, it stops your B cells. And I think, okay, let's do it, because then we get totally different repertoires. If people get rituximab, we see no change. And why is that? Because, and this is a hypothesis, or the rituximab binds to a certain population of B cells that are likely not the B cells that are producing the antibodies that are so abundant in your blood. A little bit more detail, the antibodies that are abundant in your blood, we think are produced by long-lived plasma cells in your bone marrow, while the direct responders are more the memory B cells, are the cells and so with rituximab it didn't work. We didn't see much of a change, and I was sort of disappointed, or I learned something. Now we are really looking, and for us it's not so easy to get these samples, but we are now looking at samples from donors that had a whole stem cell transplantation, for instance. And we don't have the data yet, but it's something that we want to try. And you know, it's very interesting because you can also ask, will they get an imprint from the donor? And we don't know yet.
Next question. Yeah, I have question one about the boring chromatography, because you showed the separation of the fragments, and you said that you were surprised that you can separate them. Have you tried different retention mechanisms, or I mean like different columns, and do you see that if you can get some information or pattern from that?
Yeah, luckily I also have people in my group that think like you, and they are still working on that. When I see that, I'm completely happy, and I say, let's go. But you know, we are using, it's all commercial material that we use, but there are different vendors that make different columns. Most of the work that I show is so-called MAPPAC column, works really well for us. But we do see that by changing a bit of the organic, changing a bit of the gradients, changing a bit of pH, we can get better. But again, I was surprised that we could get this, and I'm even more surprised this is on the 45 kilodalton FAP arms. For the IgG2, we don't look at the FAP arms, we look at the FAP dimers, so the FAP2 arms. They are 100 kilodalton, and we still can get this beautiful separation. So at some point, I'm not as an analytical chemist as you. I say, oh, let's be happy and let's just go. There's always room for improvement, and especially what I saw also in your group, if we could understand why they elute at a certain peak, then it would help us also in the identification in the end, I think.
Thank you. We have plenty of time for other questions. Okay.
Okay, first, thank you for a great presentation. And I would like to ask, when you were showing the repertoires, that every person has his individual, is there some connection, for example, between family members?
Very good question. So again, it's difficult to answer. We have data on 4 twins. It's interesting, from blood levels, general blood levels, we can say who are the twins. They are homozygote twins. From their antibody repertoires, there's only 4 people, or 8 people, 4 twins. We cannot say who is the twin. And I think this whole process of antibody generation is really what I said in the beginning. It's like throwing a dice, but you have 10 to the 18th possibilities. So what's the chance that you come with the same solution as me? And I think even, and that's also I didn't tell, but I want to tell now, what we see that if you get vaccinated 3 times, then at the booster, some of the antibodies you made after the first vaccination are indeed boosted. They are made again, and more of them are made. But even more so, you are making totally new antibodies against the same spike protein with your 3rd booster, antibodies that you didn't make on the first vaccination. So to me, it's sort of like your body, each time when it gets the pathogen, has this huge possibility to find a solution. And then if you look at the population, we all have these huge possibilities of solutions. And there is not a best solution. All of us makes a very good solution, hopefully. But these solutions are slightly different. That's my hypothesis from the numbers. I don't have direct evidence for that yet.
Any other question?
Who brought the butterfly?
It's spy. So if there is no other question, I would like to thank Professor Heck once again, and thank you very much for coming.